Okay, so today we're going to talk a little bit about microbial growth. Prior to this lesson, we have built up on our conversation. We've talked a little bit about the bacteria cell and other types of microorganisms. We've talked about the structure of the cell and some components that are important to the survival of the bacteria cell. We talked about the cell wall and the makeup of the cell wall. We've talked about how we can distinguish between the types of cell walls with the Gram staining. And then prior to that, we talked a little bit about just distinguishing between cell types and structures that are common to both cell types. Early in the semester, we kind of gave some historical perspectives for microbiology and how we study microorganisms. We've talked about the fact that some microorganisms are pathogenic and others are not so pathogenic, and they're actually useful for us. All right, so we're going to expand that conversation today and look at some of the ways in which we are able to grow or cultivate bacteria in the lab. What are some of the requirements? All right, so firstly, I would like to just show a quick video, all right, to recap some of the things that we've already talked about, and then we'll move into our discussion on microbial growth. So you should definitely take a few notes. Most of the things in this video we've already talked about, but it's a great recap before we move on to our next topic.
When I was a little kid, my parents would let me take my favorite set of dinosaurs into the tub, probably to persuade me into taking a bath. I remember my dinosaurs well. In fact, I used to know all of my dinosaurs. I had epic dinosaur battles. Sometimes my sister would play with them too, just a little differently. Anyway, I remember that one day, unfortunately, my favorite Stegosaurus lost his head. I mean, literally, it just popped off. And while I was determined to get my parents to fix it, we discovered that there was stuff growing in the toy. Now I realize what was actually large enough to be visible was likely mold. But at the time, my mother told me that Sergeant Stegosaurus would have to retire from the bathtub and I'd have to get a new one because bacteria had taken over. It led to two misconceptions I developed about bacteria. Number one, that bacteria were only found on or in things that had gotten contaminated somehow. And two, that bacteria are always bad, and that is why Sergeant Stegosaurus required replacing. Those are both not correct. First of all, there probably were bacteria in the dinosaur, along with the mold, but that shouldn't be surprising because bacteria are everywhere. Bacteria are found in our houses, yes, even very clean houses, and they are found outside. Bacteria colonize our skin and our digestive system. That addresses the first misconception that you only find bacteria on contaminated or dirty surfaces. And as for the second misconception, well, bacteria sure do get a bad reputation. Now, I'm not disagreeing that the toy should have been thrown out. That was a good decision. There was probably mold that was in that toy dinosaur, a fungus, but there was likely plenty of bacteria growing there with it. A growing community of mold and bacteria, not ideal for a bath toy. But we do want to mention that many times all bacteria are lumped together as a bad thing, which shouldn't happen because not all bacteria are bad. In fact, many types of bacteria are helpful for organisms and ecosystems. We'll give you some examples. Some of the bacteria that colonize your skin are beneficial and actually help keep harmful strains and other types of pathogens from growing. Bacteria in your digestive system actually help break down food, and some can produce certain vitamins. Some types of bacteria are used in producing some foods that we eat. In ecosystems, bacteria have a very important role as decomposers. Bacteria also have major roles in the nitrogen cycle to fix nitrogen that plants need. These are just a few examples of helpful, beneficial bacteria. Now, that's not to say that bacteria can't be pathogens. Bacteria are the cause of strep throat, tetanus, tooth decay, some forms of pneumonia, diphtheria, salmonella, cholera. I could go on. Antibiotics can be used to combat some of these. And while antibiotics are very important for destroying bacterial infections, we shouldn't leave out saying that some broad-spectrum antibiotics can harm some of the good bacteria as well. Also, we should mention that antibiotics do not work on viruses. Viruses are pathogens that are not made of cells at all. You can check out our video on them. Additionally, there are vaccines which can prevent many types of bacterial and viral infections. Okay, so what are bacteria exactly? In the three domains of life, bacteria encompass one of them. They come in different types of shapes, as you can see here. Some of them are heterotrophs, meaning they consume or feed on some organic matter. But some are autotrophs, they can make their own food. Plants aren't the only autotrophs. Let's take a look at this bacterium here. Bacterium is just singular, while bacteria is plural. A bacterium is a prokaryotic cell, which are generally much smaller than eukaryotic cells, like our cells. If you recall from our video, that means bacteria do not have a nucleus or other membrane-bound organelles, but you will find ribosomes, cytoplasm, a cell membrane, and nearly all bacteria have a cell wall. Like all living organisms, bacteria have DNA. Bacterial DNA, while still double-stranded, is arranged in a circular shape. Depending on the species, bacteria can also have a flagellum to help with movement, a capsule which can help give them extra protection or attachment abilities, or pili, which can help them with attaching to surfaces, including each other. Oh, and many bacterial species have a plasmid, which is basically like extra DNA. More about that later. Bacteria have some intriguing abilities that are different from our human cells that we'd like to mention. Unlike body cells, which perform mitosis and cytokinesis to divide, bacteria generally multiply even faster in a process called binary fission. Binary fission is a type of asexual reproduction, when the bacteria can easily divide to make a copy of themselves. Since it is asexual reproduction, the daughter cells would be expected to be identical to the parent cells, unless there's a mutation. Some types of bacteria do have the ability to share genetic material with each other. Remember how we mentioned that bacteria can have a plasmid, an extra copy of DNA with usually just a few genes on it? Well, bacteria can share these plasmids with each other in a process known as conjugation. The pili can be used to share this genetic information with each other. If the plasmid happens to have a gene that gives some degree of resistance to an antibiotic, this may allow the bacterium that received the plasmid to survive exposure to that antibiotic, which could be very problematic. You can learn more about how antibiotic resistance can develop over time in bacteria in our natural selection video. Bacteria can also pick up plasmids from their environment. Often when this happens, it's during a time of stress for the bacteria. In a process known as bacterial transformation, scientists can use a type of stimulus, such as a heat shock, to induce bacteria in a lab setting to pick up genetic material. There are all kinds of these uses for these genetically engineered bacteria that you can explore. Some types of bacteria can form endospores. Endospores allow bacteria to be survivors in all kinds of hostile environments: lack of nutrients, freezing temperatures, drought, just some examples. This is a reason why hospitals have to be very good at sterilization processes. We won't go through the process of endosporulation or how bacteria reactivate after forming endospores, but this is definitely something interesting to explore. Finally, some types of bacteria, along with other types of prokaryotes called archaea, can be extremophiles. Unlike our own cells, extremophiles can live in extreme environments where there may be excessive heat, chemicals that our cells would find toxic, or even radiation. Overall, we share this planet with so many kinds of bacteria that scientists continue to learn more about them every day. And if this kind of topic interests you, you may want to look into the study of microbiology. So many careers, from agriculture to the medical field to environmental work, rest heavily on the study of microbiology. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, so I thought that was a great video to sort of run back through and bring a framework for us continuing our topics. We've talked about a lot so far. Again, it's in terms of identifying organisms as bacteria, the requirements for prokaryotic organisms and how we distinguish them from eukaryotic organisms. We talked about the structure and so forth. So today we're going to talk a little bit about what it takes to grow bacteria. All right. One of the topics that she mentioned in the video was the nutritional requirements, or how they obtain their nutrients, being either autotrophs or heterotrophs. And then we also talked about the replication mechanism, like how are new bacteria cells formed. So we're going to look at some of that today. Do we have any questions before we go forward? Any more questions? Awesome. So anyway, much of the study of microorganisms depend on its ability to grow in the laboratory, right? So we're scientists, and the things that we now know and hold to be true and things that we've been able to learn is because we've been able to grow or cultivate these microorganisms in the lab, right? And so you should understand that this growth that's happening in the lab is only possible if we have what we'll refer to as a suitable media for growing these microorganisms. All right, so write this term down: culture media. All right, culture media are basically the sets of nutrients or food, if you will, that these microorganisms need in order to grow. All right, just like we have nutritional requirements as far as our diets are concerned, we have dietary supplements that we take, we eat our foods, and we do a lot of other things to get the nutrients that we need into our bodies, take vitamins and so forth. Well, bacteria also have nutritional requirements, all right, and we're going to highlight a few of those today, all right, but the general term for these nutrients that we need in order to grow bacteria are called media, all right. Media is plural, all right. A medium is a singular type of culture medium to grow the bacteria. Okay, and so this growth or culture media is a solid or liquid that's designed to support the growth of microorganisms. And so again, what it is the basic combinations of various nutrients that we know the particular organism needs in order to grow. All right, so we basically would in the lab create this optimal environment, right, for supporting the growth of our bacteria. So depending on what we're growing, we use a certain medium. We create the certain environment using the incubator with the correct temperature, pressure, and so forth, oxygen requirements, no oxygen. So we're gonna look today at the parameters that are used to grow bacteria. And if you are in lab this semester, I think, and maybe another lab or so, we'll look at and make some media, all right? So culture media is the requirement for growing and studying microorganisms in the lab. Okay, so when we talk about microbial growth, we're talking about not just growth in terms of size. Like we talk, you know, human development, there's, you know, growth and development, one of the requirements for life. Well, in this case, we're also talking about producing more, right? So this microbial growth, we see bacteria reproduce by a process known as binary fission. Okay, as the amoeba sisters mentioned in the video, basically binary fission is a form of asexual reproduction. Write that down. Binary fission is a form of asexual reproduction where the parent cell continues to divide and make more copies of itself. So it's essentially like a cloning process. Okay, it's essentially like a cloning process. Okay, this process happens quite rapidly depending on the presence of the required nutrients, depending on the environmental conditions. If the environmental conditions are optimal for the growth of our organism, then this replication or reproduction is going to happen very quickly. Okay, for example, E. coli, which we kind of use as a model organism, in bacteria, in microbiology, it divides every 20 minutes, okay? Every 20 minutes, this replication cycle, we're gonna double our population, all right? And we'll look at this concept of generation time, all right? But if we have this E. coli in the ideal nutritional media, right? And at the ideal temperature, which for this, in this case, it's about 37 degrees C, the generation time is going to be about 20 minutes for it to double its population. Okay, and what happens is that because there's this rapid reproduction, right, doubling of generations from every 20 minutes, we're doubling our population. We essentially have what we refer to as an exponential growth, right? These numbers get so large that we express them as a log of a number of cells on the growth curve. All right, so the bacterial growth curve is a very important tool in microbiology as we try to measure bacterial growth. We use the growth curve for that. All right, and we're going to spend a little bit more time next week looking at the growth curve and the information that it gives us. All right, but right now we're just painting the picture that this process of reproduction in the microbial cell, in particular bacteria cells, are a form of asexual reproduction. Okay, it requires only a single parent. And what happens is that these bacteria populations double, and they continue to double each generation time. All right. So we want to identify some factors that affect microbial growth. All right. Microbial growth is affected by two major factors, right? If you can just think about your own life, right? So we have environmental factors that influence our growth, right? And then we have sort of these chemical and nutritional requirements, if you will, for growth. All right, so we're going to look at microbial growth from the environmental standpoint as well as from a chemical standpoint. All right, environmental conditions includes the temperature of the environment, the pH, osmotic conditions. Okay, jot those down real quick and we'll come back to them in just a second. Chemical requirements, again, we're looking at some of those chemical elements that you learned about in your principles of biology class, right? We identified the chemical elements that are responsible or that are essential for sustaining life. So we know carbon, hydrogen, oxygen, nitrogen, phosphorus. These are all pertinent elements for sustaining life, right? We know that they are the building blocks for the larger macromolecules that we need, the carbohydrates, the lipids, the proteins. All right, these elements, carbon, hydrogen, oxygen, nitrogen, they are the building blocks. Okay, and so as you understand this requirement for more complex life forms, it's also true for microbial growth. Right, proper concentrations of these factors are also necessary. All right. So I mentioned the bacterial growth curve. One of the ways that we can measure bacteria growth is looking at this growth curve and actually counting our cells through spectrophotometry. So we have 3 or 4 phases that can give us an indication of how well this culture is growing. Okay, the 4 phases of the bacteria growth curve are lag phase, which is represented by this period right here. This line indicates that there is no increase in population occurring right here. But what's happening in the lag phase of the bacteria growth curve is that the cells are essentially obtaining the nutrients that they need and be preparing for division, right? But there's no reproduction happening here in the lag phase. Okay. After a while, once the optimal conditions are met, again, we've got the optimal temperature, nutrients present, and so forth, these cells are not going to start to reproduce. Okay, we're going to divide. And I mean, these populations are going to increase exponentially. All right, that's what's being represented by B in this growth curve. Okay, B is the log phase. We call it log or exponential growth phase. All right. This is when all of the conditions are optimal for these cells to continue to grow and reproduce. And we're going to continue to see this growth at a very exponential rate. At the next phase, we kind of see what we call a plateau. All right. C represents the period where the number of cells that are growing and the number of cells that are dying are now about the same. So we refer to this as a plateau or the stationary phase. Okay, stationary phase represents a period in time where the nutrients have begun to be depleted, right? And now we have death occurring pretty much at the same rate that growth is occurring, right? So we're understanding that this bacterial growth represents a period of growth over a number of hours, right? This is a culture, this is a liquid culture where we have inoculated it with bacteria cells and we're watching it grow over time. All right, we're taking samples out, we're measuring the OD in the lab, and we're observing the optical density, which is basically the turbidity of the culture over time, and we're logging these numbers and then we plot them, right? And so what we see here again at C is that we start to get a plateau. We call this phase the stationary phase, right? At this case, the cells aren't as happy as they were in the log phase, Okay, the nutrients are being depleted, things are starting to change, and we're starting to see some death occur, right? And then this D represents a sharp decline, which is basically characterized by a death phase. Now these cells are pretty much on the decline. All right, and so this is a standard representation of bacterial growth. This is pretty much what happens across the board with bacteria in terms of how they grow. Okay, now again, there are a number of factors that influences the growth of these bacteria, right? But we know we can sum up and be able to explain what's occurring. This is a basic schematic of what's happening, okay? During A, which is the lag phase, we say lag because there's no growth happening here. It just kind of looks like a lagging process. And real quick, I forgot to tell you the axes. The x-axis is time. Okay, this is how much time is being elapsed, and the y-axis are the number of cells that are being produced. Okay, and so we see here a lag phase. There's no increase in the number of cells. It's just that time is passing, and these cells are basically preparing for division. Okay, that's lag phase. Log phase, we have the exponential growth, where these cells are reproducing by way of binary fission, and the generation times are doubling. C is the stationary phase, where we're starting to see our plateau. We don't see a whole lot of exponential growth anymore. In fact, the rate of growth and depth are starting to balance each other out. And then we have our period of decline here over in D. All right, and so we will spend a little bit more time next week with the growth curve. Okay, but I guess you're like, okay, so why do we study the growth curve? It's very important in us studying microorganisms. All right, it helps us to understand how microbes grow under different conditions. Again, we can manipulate the environment of the organism, and we can study and collect data and make observations based on the things that we're changing and manipulating. Okay, we can adjust the growth conditions. We can better understand these organisms when we're studying as we're making these manipulations. Okay, so it helps us to understand how these microbes grow under different conditions, right? If there's a nutrient depletion, is there a lack of oxygen, is there a lack of carbon? We can see what happens under these varying conditions. Okay, and that is important for us. We study these growth curves also because they help us understand how pathogens grow in our bodies. So again, we can create these optimal environments that sort of mimics what a host would look like, what our bodies look like. We can mimic that in terms of pressure, pH, temperature, right? We can create those environments, again, in an effort to study and sort of extrapolate that this is what would happen if a pathogen were to invade our bodies, right? And then it also helps us to study the effect of different chemicals and other conditions in terms of osmotic pressures, different temperatures, and so forth. So we get to study all of the parameters that we may be interested in that we know has an effect on bacterial growth, right? So we can set up any environment that we wish almost in order to examine what happens to the organism under these conditions. Okay, so we can sort of predict then what would happen if we grew something like E. coli, which is one of the most studied microorganisms, and it's sort of a model for us for what happens when we're growing gram-negative organisms. We can predict, you know, what the growth curve would look like under certain temperatures for so many hours and so forth, because we have a standard that we now know due to us studying the growth curve. What questions do we have about this? Any questions? Awesome. So let's look at some factors that affect microbial growth. Let's start with physical factors that affect bacterial growth. Jot these down real quick. We can really quickly identify bacteria based on how they are responding to temperature. All right. Physical factors, temperature. We can identify these organisms as either mesophiles, sacrophiles, and thermophiles based on their temperature preferences. Okay, I do want you to know these ranges of temperatures and how we categorize these bacteria. right? Mesophiles are those bacteria that grow best in moderate temperatures. So we're talking from about 25 to 40 degrees, all right? And this is where most of our pathogens are gonna lie in this range, right? About 37 degrees C is gonna mimic human body temperature, all right? So most of the organisms that we grow in the lab, most of those which are potentially human pathogens are gonna be our mesophiles, right? They want to grow in somewhere around 37 degrees. All right. And most of the other organisms that we work with in the lab are going to fall somewhere in this range, and they grow from about 30, 32, 37 degrees. Psychrophiles, these are kind of on our extremes now. These are adapted to survive and grow at cool temperatures, okay, in the refrigerator. So we're talking about below 25 degrees C, okay? These are our psychrophiles, cold-loving organisms. They have adapted such that they can survive in very cold temperatures, all right? Keeping in mind that most organisms don't prefer to be there, okay? Most microorganisms, at least the ones that we study and come in contact with in the lab and for the purposes of disease, human diseases and so forth, they are going to fall in that mesophile range. So do make a note of that. And then those, the last group are thermophiles. All right, so we're still looking at temperature here. Thermophiles are those organisms that have adapted to grow in very high extreme temperatures. Okay, so we're looking at over 60 degrees. All right, the Thermus aquaticus, for example, we've extracted our enzyme Taq polymerase from this particular bacteria, right? And it's a heat stable enzyme that we utilize in the lab quite frequently. All right. And so these thermophiles can survive in extreme heat and high temperatures. Saprophiles are those that are going to survive in cold temperatures. And mesophiles are going to kind of be in that middle, looking for a more mild temperature range of about 25 to about 40 degrees C. Okay. So you should be able to sort of identify and distinguish between microorganisms in terms of these physical factors that we're looking at here, which is temperature is one physical factor that affects bacterial growth. All right. Another physical factor that affects bacterial growth is pH, right? We know that the pH is a measure of the acidity or the alkalinity of our media or whatever we're growing our microorganisms in, right? Most bacteria are going to grow kind of around that neutral pH. Make a note of that, right? Most bacteria are going to grow at or around a neutral pH from about 6.7 to 7.5, all right? A neutral pH is around 7. All right, so pH is another physical factor that influences the growth of microorganisms. Fungi, for example, is another microorganism that prefers to grow in an acidic environment under slightly acidic conditions. So pH from about 5 to 5.5. And I don't know if you remember, but you learned about the pH scale in your Biology 103 class, right? So the pH scale is a measure of how acidic a solution is, right? And it ranges from 0 to 14. Right? So solutions that are anywhere from 0 to about 6 are considered to be acids, right? And then solutions that have a pH from greater than 7 to 14 are said to be basic or alkaline. And then solutions that have a pH at or around 7 are said to be neutral, right? So those solutions include things like water, our blood, right? Those are neutral solutions, right? And so fungi are a type of a microorganism that can grow in slightly acidic environments. Okay. Helicobacter pylori, H. pylori, is a bacteria, which is certainly a human pathogen, has unfortunately adapted to our acidic environment in our stomach, and it can survive. All right. H. pylori is very problematic. It causes stomach ulcers, and it's also been linked to causing cancer. Okay. So we're still looking at some physical properties that influence bacterial growth. So we looked at temperature, we looked at pH. All right. Yeah, so other factors, osmotic pressure. All right, so we're talking about the relative salt concentration, all right, of a solution. All right, and we know depending on the concentration of salt, these solutions can have varying effects on the cell in terms of the cell taking up this saltwater solution. All right, so hypertonic solutions are going to have a higher salt concentration, and it's actually toxic for bacteria. It's going to stop bacterial growth. Okay, hypotonic solutions are going to have a lower salt concentration, increased fresh water. There's going to be a net movement of water into the cell, right, and it can essentially lead to lysis or rupturing of the cell. Isotonic solutions are those that are kind of neutral, if you will. There's going to be an equal net salt concentration in terms of the water and salt concentration, or solute. We will prefer an isotonic solution at all times, an isotonic environment for maintaining cell stability. All right. And so, right, so this osmotic pressure, again, can play a role in bacterial survival or growth. Okay. So we have temperature, we have pH, we have osmotic pressure, all as factors that can influence bacterial growth. All right. So those are physical factors. Let's look real quick at some chemical factors. Jot these down. All right, chemical factors that affect bacterial growth. All right, we're looking at nutritional requirements here now. All right, how microorganisms acquire the nutrients that they need. We know we need our carbon, we need hydrogen, oxygen, nitrogen. We need a range of chemicals for maintaining that homeostatic environment. All right, let's look at how we get a carbon. All right, organisms can be distinguished based on how they get their carbon sources, based on their sources of carbon. All right, just like other organisms or animals or plants that have been identified as either autotrophs or heterotrophs, we can also do that for bacteria. Okay, there are autotrophic bacteria, which are producers. All right, they undergo photosynthetic processes in order to produce glucose. They can make their own foods. There are some, put a nice asterisk by the word some. Not all bacteria are going to be photosynthetic. Okay, cyanobacteria is an example of one that is autotrophic in nature, can make its own food. Right? We know this process of photosynthesis has the reactants carbon dioxide and water in the presence of sunlight energy in order to produce glucose and oxygen, right? In contrast, heterotrophs require food from an external source. We have to consume our food, all right? And so bacteria also must do this, all right? And most microorganisms are going to have this heterotrophic nature. They're going to require an external consumption of food or absorptive nutrition, okay, for obtaining the nutrients that they need. All right. We have our chemoautotrophs, which kind of have a more unique metabolism. They're using chemical energy from inorganic molecules for the production of carbon. All right. So these chemical factors that we need, carbon is a major source, right? Another one, oxygen. Absolutely, we can distinguish bacteria based on their oxygen requirement, okay? Write these 4 categories down. We have obligate aerobes, obligate anaerobes, facultative anaerobes, and microaerobes, all right? This is based on the presence or the necessity for oxygen. Obligate aerobes are organisms that absolutely require oxygen in order to survive. There's no ifs, ands, and buts about it. Obligate aerobes must have oxygen in order to survive. Okay. Obligate anaerobes. So we have aerobes and anaerobes. Okay, that prefix "an" in science means without. Obligate anaerobes are absolutely, oxygen is lethal for them. Okay, it does not require oxygen to survive. Okay, well, not only does it not require it, oxygen can be potentially lethal for it. No oxygen. It requires an atmosphere with no oxygen present. Obligate anaerobes. Okay. Facultative anaerobes. These are organisms that don't necessarily have they can grow in the presence or absence of oxygen. Okay. Facultative anaerobes can grow with or without oxygen being present. Okay. So it's not a requirement, but it also doesn't cause any problems if it is there. Okay. The microaerophils grow best in low oxygen. Okay. A little bit of oxygen present is ideal. Okay. But not a completely aerobic environment. Okay. So low oxygen and high carbon dioxide. All right. So other chemical requirements, carbon, absolutely. Carbon sources, we can distinguish organisms or bacteria based on their source of carbon, based on their oxygen requirement. Okay. Again, for some organisms, particularly those that are facultative anaerobes, the presence of oxygen can be problematic. Okay, oxygen can be toxic. You know, it oxidizes and sort of destroys other chemicals that are present. Okay, aerobic organisms have enzymes and systems that are like these built-in mechanisms to handle the presence of oxygen. Okay, so one example is this enzyme SOD, the superoxide dismutase is an enzyme that chemically alters oxygen, or these free radicals that are produced in order to make the environment more tolerable for bacteria. Okay, catalases are also enzymes that converts hydrogen peroxide to oxygen in water. And so again, aerobic organisms have the physiological capability to process or utilize oxygen that might be present in the environment. All right. Other chemical requirements. Okay, nitrogen. All right. We know that nitrogen is a major component in amino acids. All right. Amino acids are essential for the production of proteins. This does not change in bacteria. Bacteria undergo transcription and translation just like eukaryotic cells do. The production of proteins are absolutely essential for the survival of the bacterial cell. Okay. Nitrogen is also found in our nucleic acids. Again, bacteria have genetic material, DNA and RNA. It is essential for the survival of that bacteria cell. All right. So the presence of nitrogen is essential for microbial growth and development. Hydrogen. All right. We know that hydrogen is found in all biological molecules. Those carbohydrates, fats, proteins, nucleic acids, all of those biological molecules that you learned about in Biology 103 has hydrogen as one of its building blocks, all right, in those monomers. Phosphorus found in our nucleic acids, ATP, which is our source of energy, those phospholipids, which make up the major structure of that cell membrane. We talked a lot about the structure of that membrane. We know that phospholipids are a very unique type of lipid that's found in the cell membrane, all right? Phosphorus is absolutely essential for microbial growth. Sulfur, again, we're going to find these on some of the side chains of amino acids. And again, we need these. The amino acid, whatever organisms require these amino acids to be present, you're going to find sulfur. Okay. Trace elements. I think we may have mentioned trace elements a little bit, but if we haven't, we establish those chemical elements that are found in great abundance in living things, right? Our carbon, hydrogen, oxygen, and nitrogen. Well, there are others that are also very essential, though they are found in really small amounts. And we call those trace elements. Okay, trace elements are also an important factor in microbial growth. Okay, inorganic elements that are needed in very tiny or minute amounts or concentrations, right? So zinc, manganese, cobalt, these can all be requirements for microbial growth. All right, so do make a note of those. All right, other organic factors, we can have other vitamins that are required for some bacteria, other coenzymes that are required, special types of media. We can pretty much formulate media, growth media, to contain whatever substances, supplements, nutrients that we need for our microorganism to grow. Okay, and so, like, for example, fastidious organisms are quite unique and they have additional media requirements. Okay, and we can formulate those in the lab. Okay. Right. So some organisms are very hard to culture. We cannot necessarily culture everything in the lab, although that would be quite ideal. But there are some that have very strict intracellular or host requirements that makes it a little bit difficult to culture them in a laboratory setting in vitro. Okay, so they require a host cell in order to actually cultivate them, eukaryotic cells. All right. Let's stop right here. We'll pick up with some metabolism on next week. We're just about out of time for today.